Good afternoon, everyone, and thank you for the very kind invitation to having me out here. Thank you to our Israeli colleagues for making the trip here, although I would say that I was looking forward to visiting Israel myself. This is actually the third visit, or the third invite to Israel, whereby the meeting was canceled. So I was really looking forward to making it this time.
Next year.
Next year, right. So just a few disclosures I need to mention, and perhaps the first one is probably the most relevant. Tempus AI, I serve on their scientific advisory board, and I own equity in Tempus AI, which is a precision oncology company. What I will talk about today is really our efforts in the context of cancer in terms of developing short-read sequencing-based technology, so generally Illumina short-read sequencing technologies, how that can be used in the context of clinical sequencing of advanced cancers, and we've been heavily involved in using this general approach for advanced or metastatic cancers, and how, looking into the future, how in a clinical context, long-read technologies such as the Oxford nanopore long-read technology could potentially complement Illumina short-read-based technologies in terms of precision oncology workflows. And that's what I'll be presenting today. And I'll give you a little bit of background, and this sort of connects in well with Eric's introductory talk, except really heavily focused on cancer genomics in general and some of our work using short-read technology-based approaches. So my group in about 2009, 2010 established the MyOncoSeq program, or the Michigan Oncology Sequencing Program. And so this was in the early days of next-generation sequencing and its use in terms of advanced cancers. And we established this general idea that one could basically comprehensively sequence a tumor's DNA and RNA to define molecular alterations that one could then potentially ascribe to in terms of clinical trials or off-label use of approved agents, or that this technology was maturing to a point whereby we could think about bringing this to cancer patients. And so this is actually one of the first of its kind clinical sequencing programs established. And the reason that we got into this was really because of our discovery efforts around discovering recurrent gene fusions or translocations across an array of epithelial tumor types. And we were developing bioinformatic approaches as well as NGS-based transcriptome approaches to nominate gene fusions in epithelial tumors. And so this is the general sort of precision medicine workflow that we established in about 2010, 2011. This basic idea that advanced cancers or metastatic cancers you'd take a tumor biopsy, compare that to a matched germline sample, generally a buccal swab or a blood sample, carry out integrative sequencing, bioinformatics, computational analysis. This would then be, in the early days, all of the cases were basically presented at a precision medicine tumor board, now probably more commonly called molecular tumor board, and we established one of the first of its kind precision medicine tumor boards, and then these results could then be returned back to individual patients. So we established this, again, using comprehensive sequencing-based approaches in the early days, but now this has been really operationalized by many commercial entities as well as cancer centers that are really focused on targeted, actionable panels in the context of cancer. So when we sort of prototyped this general approach, we just showed that we could essentially use this approach across 30 different cancer types, as well as 30 different sites of biopsy, primarily because we're not only sequencing DNA, but we're sequencing RNA, so you get sort of an epigenetic state as well as the tumor microenvironment when you're covering RNA sequencing in conjunction with DNA sequencing. But one of the early observations that we made in this assessment was the fact that about 12% of advanced or metastatic cancers across different lineages actually harbor pathogenic germline alterations. So these are germline alterations relative to somatic alterations that were very much enriched in patients with advanced metastatic cancers. And so, again, these are sort of in the early days, the first 1,000 patients that we sequenced. Only a small fraction of them received sequencing-directed therapy, and out of those patients, about almost 40% seem to have some level of clinical benefit from knowing what the molecular profile was for that patient's tumor. And again, this is early days where there weren't as many clinical trials available, and not as many drugs were necessarily approved in terms of the first 1,000 patients that we sequenced. We've now sequenced well over 7,500 patients with advanced or metastatic cancers. So from having this clinical sequencing program active at the University of Michigan, we've actually come across a number of intriguing observations that I sort of list here, and I'll just walk through a few of these particular examples. And these were actual patients that we enrolled in our clinical sequencing program that is, again, run in a CLIA-CAP laboratory and so forth. So we were able to really define the gene fusion landscape of prostate cancer. Beyond the initial fusions that we discovered, the TMPRSS2 ERG fusion, we were able to identify a number of other ETS transcription factors that hit other subsets of prostate cancer, FGFR fusions, R-spondin fusions, RAF kinase fusions that define mutually exclusive subsets of prostate cancer. So prostate cancer is indeed a gene fusion-driven cancer type. We were able to identify the NAB2 STAT6 gene fusion as a pathognomonic gene fusion for solitary fibrous tumors. So this tumor is, of course, now defined by this particular gene fusion that's the driver of this disease. Using, again, these technologies, we were able to identify FGFR gene fusions across an array of epithelial cancers, but in particular, we found an enrichment in cholangiocarcinomas. And this then sort of laid the groundwork for FGFR inhibitors to be approved in FGFR fusion-positive cholangiocarcinomas. Likewise, our group and Sarat Chandrapati's group at Memorial Sloan Kettering discovered a enrichment of activating ESR1 mutations or ER mutations in hormone-resistant breast cancers. At a very high percentage of patients with metastatic hormone-resistant breast cancers harbor activating mutations of ER. And this then, again, sort of laid the groundwork for a most recent FDA approval of selective ER degraders or SERDs in the context of ER or ESR1 mutated breast cancers. In terms of sort of multi-institutional team efforts, I was fortunate enough to team up with Charles Sawyers at Memorial Sloan Kettering and lead a Stand Up to Cancer Prostate Cancer Foundation Dream Team focused on metastatic prostate cancer, where we were basically trying to proliferate these precision oncology approaches across institutions in the US as well as in the UK, led by Johan de Bono. We were able to define the first landscape of genomic alterations in metastatic castration-resistant prostate cancers. A number of key alterations were discovered in this particular study, different pathways that are altered and so forth. But perhaps the most profound finding from this particular study was that up to a quarter of patients with metastatic prostate cancer harbored defects in DNA repair pathways. And this was certainly interesting because this was certainly not known in the context of metastatic prostate cancer at this time, but the fact that these pathways are quite actionable was important for these patients. This then, along with clinical trials led by Johan de Bono, really led to PARP inhibitors being approved for homologous recombination deficient prostate cancer. So this is upwards of about 14% of patients have defects metastatic prostate cancers have defects in BRCA1, BRCA2, and PALB1. Likewise, we also discovered that upwards of 3% of metastatic prostate cancers harbor defects in mismatch repair genes. And these patients, of course, respond to immune checkpoint inhibitors, which were already approved in the context of mismatch repair. One of the other genes involved in DNA repair pathways that we also discovered in this initial study was that approximately 7% of patients harbored defects in CDK12. And our group, as well as Matthew's group, published that CDK12 is biallelically altered in about 6 to 7% of metastatic prostate cancer. These patients have an increased gene fusion burden relative to other molecular subtypes of prostate cancer. So when a patient has a biallelic loss of CDK12, this leads to extensive tandem duplication that occurs across the genome. These tandem duplications then create gene fusions, which lead to genes essentially being expressed out of frame, creating neo-open reading frames or neoantigens that emanate from these fusions that then presumably induce immune cell infiltration. And this is, and we certainly see T cell recruitment in the context of CDK12 mutant prostate cancer. In terms of another transcription factor that's involved in prostate cancer, this includes the pioneer transcription factor FOXA1. We were able to use this short-read sequencing technology to define the various classes of FOXA1 alterations that occur in prostate cancer, but in particular, we were able to discover the gene rearrangements or tandem duplications that occur at the FOXA1 locus to trigger overexpression of FOXA1 in a subset of prostate cancers. So now transitioning to basically this idea, so that was a lot of sort of short read sequencing technology. So we really wanted to address the question, and can we begin to take advantage of some of the longer read technology that's being implemented, in particular the Oxford nanopore long read sequencing approach, and to see if this approach could complement short read sequencing based approaches in terms of precision medicine workflows. So this particular technology takes advantage of these nanoscale protein pores that basically nucleic acids can flow through and cause changes in electrical current that then can be detected in terms of base differences. And this offers primarily the ability to carry out direct sequencing without the need of PCR amplification, but also very long reads relative to Illumina-based technologies. And so just a bit of background in terms of next-generation sequencing-based approaches. Certainly we have the first-generation Sanger sequencing. Then there's, of course, the massively parallel second-generation sequencing by Illumina. And then moving on to the topic of the rest of my talk is around the third-generation long-read sequencing. And in particular, I'll focus on nanopore-based, Oxford nanopore-based sequencing, but there are, of course, other long-read technologies available. And in terms of the aspects that the nanopore long-read sequencing enables, this includes very long, ultra-long reads exceeding 100 kb. Sometimes you could essentially sequence an entire genome of certain species. Structural variant detection is probably what it's best known for, and being able to detect large insertions, deletions, inversions, and translocations. You can take advantage of this technology for haplotype phasing in terms of allele-specific analyses, and I'll give you some examples of these. As I mentioned earlier, this is a technology that allows for direct detection of DNA modifications without the need for bisulfite treatment. So essentially you can monitor methylation across the genome. Again, no PCR amplification in contrast to the Illumina-based technologies. There are also abilities to carry out real-time adaptive sequencing, where you can basically tailor the time frame as well as the flow cell usage for a particular indication. And then, of course, there's thoughts that this technology can be cost-effective and scalable. There are, of course, certain disadvantages to the long-read technology as well, but I'll be really focusing on it as a complement to the short-read technology, and not necessarily a replacement. So this approach has been studied and investigated by a number of groups in different clinical indications, generally in sort of specific areas, critical care, and in trying to carry out the sequencing in a rapid fashion. But no one has really focused on trying to bring this technology to the context of precision oncology, and how do we complement short-read technology with these approaches. So we talked considerably about the short-read based technologies here, and so the idea is can cases that end up being cryptic in the context of short-read technologies, can the molecular mechanisms be deciphered with this nanopore long-read technologies, which then affords the ability to look at complex SVs, haplotype phasing, viral integration, HLA typing, DNA methylation, and so forth. And how can one incorporate long-read technology in concert with the short-read technology in terms of precision medicine workflows? One thing that we were able to do with this technology, and now we've sequenced, I would say, over 200 advanced cancer patients with the long-read sequencing technology. And what we find is that we can actually carry out the approach in a clinically relevant time frame. I would say that generally we can carry out up to the point of sample collection to turning around a report within 10 days. But comfortably, we can do this within 2 to 3 weeks. So certainly the technical approaches, as well as even the bioinformatics analyses, can be operationalized in a clinically relevant time frame in the context of oncology. So this is just a graphic of the bioinformatics and computational pipelines that we've assembled to carry out these analyses, and these are generally a compilation of open source algorithms as well as some algorithms that we've developed in-house, but to work together in order to carry out these analyses in a regimented time frame. So one of the clinical questions we try to address is, can this technology be used in resolving cryptic variants in a cohort of advanced cancers with signs of genomic instability? And in particular, we were interested in mismatch repair deficiency, focal tandem duplications, perhaps due to CDK12 inactivation, and homologous recombination deficiency, which could be due to BRCA1, BRCA2, and so forth. So in this cohort of patients, short-read sequencing actually does a pretty good job. About 70% of these cases, you can actually identify the definitive mechanism as to what caused that genomic instability in the context of short-read sequencing. But then that leaves about 20 to 30% of cases whereby short-read sequencing ends up not being able to identify the key mechanisms of that genomic instability. So here I'm just summarizing a subset of our long-read cohort, and here you can see that even in our cohort, about 70% of the cases were certainly deciphered with short-read technologies, and this includes a cohort of prostate, breast, and ovarian cancers. But about 30% or so, 20 to 30%, long-read technology could actually complement short-read technology to actually define the mechanisms of genomic instability. And here I'll just flash through some of these key cases in our cohort. These are cases that long-read technology basically was able to define the mechanism, and these are mismatch repair deficiency cases. And generally, these were long deletions, long translocations, and promoter methylations and so forth that really help define the biallelic mechanisms of these in the case of mismatch repair deficiency. In terms of focal tandem duplication, we were able to detect large tandem duplications in CDK12, deep deletions, complex rearrangements, and large translocations, again using the long-read technologies. In homologous recombination deficiency, what we found was that we were really able to take advantage of the methylation aspects of the nanopore technology in silencing genes like BRCA1. We're able to detect methylation events in, for example, BRCA1 and C, essentially the biallelic hits that are occurring in these cases that would, of course, be invisible in the context of standard Illumina short-read technology focused on DNA. So now just to run through a few examples sort of outside of genomic instability and how we've been able to take advantage of these cases. And these are, again, actual patients that enrolled in our clinical sequencing program. We were able to take advantage of the haplotype phasing aspect of long-read sequencing to really gain insight into the origin of whole genome loss of heterozygosity. So this patient here was basically a patient with androgenetic biparental mosaicism in terms of a tumor that was identified, multiple tumors that were identified in this particular patient that we were able to ascribe to an androgenetic biparental mosaicism, and we were able to really take advantage of the long-read sequencing to facilitate haplotype specific methylation analysis. In the second case, this is an ovarian teratoma, where we were able to essentially see that the mechanisms that were occurring in this particular case were post-meiosis II, and we were able to detect crossovers of DNA, crossovers of the haplotypes of DNA, and we were able to see that specifically in the teratoma DNA sample. Again, sort of providing the mechanistic insight as to how these patients exhibited whole genome loss of heterozygosity. In terms of resolving cryptic germline events and phasing variants with long-read sequencing, we were able to see retrotransposon ALU elements that were inserted into genes like BRCA2. We were able to see long insertions that could affect splice acceptor sites. The other aspect of the phasing of variances is that in certain genes, these mutations need to occur in a cis fashion. So using long-read technology, we can essentially monitor, for example, alterations in the notch gene, which generally we predicted to be cis in nature, on the same allele, but using short-read technology, you can't really necessarily detect that. But for example, with Notch1, we could essentially see mutations in the juxtamembrane domain, as well as in the pest domain happening on the same allele using, again, the long-read technology. And then one interesting case that we were able to identify is this 3-year-old girl that had a adeno-associated virus gene therapy, and we were able to actually describe the mechanism for how that tumor was generated in this patient that had a spinal tumor. And this, again, this 3-year-old girl was received an AAV-based therapy, and this actually represents the first documented case of tumorigenesis caused by an adeno-associated virus. And it's using the long-read technology, we were able to monitor the insertional immunogenesis. So this would be coming from the gene therapy vector, and we were able to see that it was that elements from the vector, including the promoter and enhancer elements, were inserted into the intron, the first intron of GLI1, that then led to overexpression of the GLI1 transcription factor, which is part of the sonic hedgehog pathway, and be the reason that this particular patient developed this cancer. And this is actually kind of somewhat of a shocking finding, because it's really thought that these adeno-associated viruses generally function in an episomal fashion. So this is actually showing that the virus actually integrated into the human genome, or at least in the context of this tumor. And so this was actually reported to the FDA. And then just a last slide in terms of, since we have a number of talks around AI, I think they might be tomorrow or in the later sessions. I wanted to also just give you sort of what we're looking at in terms of how pathology will play a role in terms of AI-guided precision oncology, with the idea that essentially integrating clinical outcomes data, genomic alterations, whether it be short and long-read sequencing, taking advantage of the whole slide imaging and the various morphologic features one can get from a standard H&E slide, coupling that with advanced spatial transcriptomics and so forth, and then we're now in the position now to really create multimodal models that one can develop in terms of training and testing, validation, and so forth. And we believe that this general approach will certainly impact AI-guided precision oncology into the future. And then finally, just to close with some take-home points in terms of integration of long-read sequencing and precision medicine workflows, this really addresses, I would say the long-read technology addresses blind spots of short-read sequencing, including structural variations, methylation, haplotype phasing. They're very useful in defining the mechanisms of genomic instability, and it actually can boost the diagnostic resolution by about 20% from where you were with using standard conventional Illumina-based short-read technologies. Overall, these long-read approaches, this integrated approach of using long-read and short-read approaches, really improved the actionability to an additional 20% of patients with advanced cancer. I showed you the demonstration of the first human case of an adeno-associated virus, where the virus had an insertional mutagenesis event to trigger the disease. This tool is very useful for cryptic germline as well as compound variants, and certainly in terms of haplotype phasing. And these are the individuals that really led this work in the lab, and those in red would receive particular credit, I would say. And then just a last slide here. Thank you for your attention.